All right, we are going to cover the sense of vision in this video. So this is another of our special senses. So we're going to start off by talking about some of the structures that protect the eyes because this is a really important sense for humans. Obviously, we can survive okay without it, but it is really helpful. So structures that protect the eyes are going to include the bony orbit. So this is the eye sockets of the skull. And then our next structures are going to be the two eyelids and tarsal glands, and then something called the lacrimal apparatus. So first off, let's talk about the eyelids. So we have an inner lid. This is a very thin membrane known as the palpebral conjunctiva. So you've probably heard the term conjunctiva before. And that's usually what people refer to this as. This is going to help connect the actual eyelids to the eyeball so that they are attached to each other. And there is a thin layer of ocular conjunctiva that is where that connection happens. So this is a very continuous or solid layer on the external anterior surface of the eye, except for the cornea. So this does not cover the cornea, and this is going to be superior to the white part of the eye, or the sclera. All right, and we'll see a graphic of this here in just a little bit. And this conjunctiva is going to have blood vessels and nerves, which will help support the sclera, which does not have blood vessels in it, so it's avascular. And it's also going to help detect any foreign particles entering the eye, like dust and possibly pathogens. So here's that graphic. So here's the eyeball. We've got the cornea sitting over that front part. Sclera is the whites of the eye. And then we've got the ocular conjunctiva, which is kind of fused together with that sclera. And then the other conjunctiva layers out here, kind of covering some of that muscle and fatty tissue. So we do have a lot of adipose tissue surrounding the eye for a lot of cushioning. So we really need that. That's really important to have that cushioning. I almost said conditioning. Don't know why. It's the last week of the semester. I'm going to blame it on that. So that conjunctiva, again, has blood vessels. The sclera doesn't. So the actual sclera itself looks like it might have blood vessels on it that can get bloodshot, so red eyes, but it's actually part of that conjunctiva. There are bacteria and viruses that can infect the conjunctiva, which then gets very red and swollen. So this is commonly referred to as pink eye. So the official name for pink eye is conjunctivitis. So anything that ends in itis in the medical community is inflammation of something. So tendinitis is inflammation of a tendon. Conjunctivitis is inflammation of these conjunctiva. Next up is the lacrimal apparatus. So it's responsible for the production and drainage of lacrimal fluid, more commonly known as tears. Tears are really important because they're going to help reduce friction of our eyelids as we close and open our eyes, and it helps continuously clean that anterior surface of the eye, as well as preventing bacterial infection because our tears do contain a lysozyme in it. So this is an enzyme that can break down bacterial cells. So our tears are produced by the lacrimal gland, located in a depression on each of the orbital sockets. So this was the nasolacrimal canal that we covered in Unit 2 with the skull. There are small openings called lacrimal puncta that are going to drain that lacrimal fluid into some channels. These are the canaliculi, lacrimal canaliculi. There's a superior and inferior one. And from there, it enters the lacrimal sac, and then through the nasolacrimal duct, through that passageway in the bone, the canal, and this all ends up in the nasal cavity, mixing it with mucus, which will trap more of that dirt, debris, and pathogens in it to eliminate that from our body. All right, so here's our lacrimal gland up here, kind of at the lateral and superior side of the upper region of our eye, so just kind of under where the eyebrow would be. Lacrimal gland is going to produce all of those tears, and we do make these consistently, again, to keep the eye lubricated and prevent scratching or damage to it in any way. But obviously there are some scenarios where it's going to produce a significant amount of those tears, of that fluid, usually through either emotional situations like really extreme sadness or anger even, but also in cases where maybe you're driving down the freeway or a road with your windows open and all of that wind coming in is going to make your eyes water a little bit. Super dusty conditions make your eyes water and stuff like that. So those are all cases where we're going to have increased production of that tear fluid from this lacrimal gland. And then here are the punctums, superior and inferior, to help drain that fluid. Here is a canaliculi, so the superior and inferior canaliculi, lacrimal sac, and through that nasolacrimal duct further down into that nasal cavity. All right, let's talk about movement of the eyes now, because we need to be able to move our eyes around. We have 6 muscles. These are called extraocular muscles, so outside of the eye. They are innervated by cranial nerve number 6, the abducens nerve, so that's not one that you needed to learn for lab, and neither is the trochlear or ocular motor nerve. You don't need to learn any of those for lab, but they are there. And so we've got these 3 nerves that are controlling the eye muscles, the muscles that move our actual eyeballs. So with 3 different cranial nerves controlling that kind of tells you how important it is to have those eye movements. So you don't need to know the names of any of these muscles in here, but you can see they're going to do different jobs. So we have elevation of the eye, so being able to look up. Adduction of the eye, so add is closer to the midline, so this is going to be turning an eye in towards the nose, so looking kind of across to the side. One of your eyes is going to have this adduction movement, the other one would have abduction, so moving away. Then we have depression of the eye, so looking down, and then we've got some other muscles here, the oblique muscles that can both abduct, depress, and internally rotate for the superior, and then abduct, elevate, and externally rotate for the inferior one. So here's what they all look like. So from this side view, so this is the lateral view. So we've got some muscles here. You can imagine that this muscle right here, if that contracts, it's going to pull back on that tissue and that'll turn the eye upwards. Similar with this one down here, if it pulls, it's going to turn the eye downwards. So you can kind of see where these muscles are attached to, both on the eye and the tendon back here at the back of the eye socket. Kind of indicates what movement they're going to do. So this muscle is going to be connected back here, so if it contracts, it's going to pull the eye to the side. All right, and then we've got another one back over here. All right, and then the oblique muscles, as you can see, are kind of up and lower from the rest of these muscles. So the superior one is attached to the structure up here, the trochlea, so there's some more tendons and stuff up here, and then there's further tendons connecting down, so that's how it's able to move the eye in different directions. And then this inferior oblique muscle is connected to the eye, and then the front part of the bony eye socket, also allowing those multiple movements. So here's that inferior oblique muscle right here that's going to be able to do some additional movements. All right, let's get into some additional structures of the eye. Many of these were already covered in lab, so I'm not going to go into a lot of detail about those. So that fibrous tunic or layer includes the white sclera and the clear cornea over the front of the eye, over the iris and pupil. The vascular tunic, so this is more connective tissue, is the choroid and ciliary body attached to the lens, as well as suspensory ligaments and the iris itself with that pigmented layer. And then we have 2 layers of smooth muscle. These are called the pupillae. So we have a sphincter and dilator that will open and close the pupil. And then we have the neural tunic, also known as the retina, which is containing the rods and cones, which are photoreceptors. And then, like we covered in lab, we have the anterior and posterior cavities. So the anterior cavity is the cornea and lens, so that'll include the iris filled with aqueous humor. Posterior cavity is everything behind the lens filled with vitreous humor. So here's that nice graphic of the eye. So again, I don't want to go into a significant amount of detail here since a lot of this was already covered in lab. And so you should be familiar with a lot of these structures again. So here's that optic nerve, lots of blood vessels coming in, and then more vessels will come in from other parts of the body, not just through with that optic nerve. So where that optic nerve comes in creates a blind spot, so that's our optic disc. I'm going to try to remember with this week's module to post a resource for how you can check and find where your blind spot is on your own at home. It's actually a really easy test, and it's kind of cool and a little bit trippy to actually identify that blind spot, and you just don't realize that you have a blind spot because your other eye is going to make up for that blind spot, so its vision will cover that blind spot. So you would potentially notice that blind spot if you have impaired vision in only one of your eyes. So here's those iris muscles. So we're going to be able to constrict and shrink down, close that pupil. That's going to reduce how much light is coming into the eye. So when you walk out in the bright sun, this is what's going to happen, is that constriction is going to happen. Your pupil's going to shrink because you don't need as much light coming into the eye because the environment is supplying an ample amount of light. Whereas when it's dark, when it gets evening or super early morning before the sun's up, then we are going to expand that pupil and open it up so that we are allowing more light to come into the eye so that we have a little bit better vision. But humans in general, we don't have great night vision the way other animals do. So there are limitations to this, but this is going to help control how much light is coming into the eye. All right, let's get into actual vision now, and this involves the retina. So the retina has several different layers of cells all connected to each other. So from the deepest layer to the most superficial, so most deep are photoreceptors called rods. This is for light vision, so sensing light, and then cones, which are for our color vision. Then we have horizontal cells. All right, so the horizontal cells are going to help with processing. And then we have bipolar cells. These are going to have some of the neurotransmitters from the photoreceptors released onto them that then connect to the retinal ganglion cells up here on the superficial layer. We have amacrine cells. These are going to work with the horizontal cells to help with that retinal processing before we have the electrical or action potential produced by this final layer, the retinal ganglion cells or RGCs. So their axons are going to all gather at the optic disc and leave the eye as the actual optic nerve. All right, so this is what is transferring that final electrical impulse to the actual brain. So at the dead center of the retina is a very small area known as the fovea. Central fovea or fovea centralis are other names for it. That's what was given in your lab guide. So at this point, the retina completely lacks all of these supporting cells and blood vessels and contains only photoreceptors. So at that fovea, we don't have the horizontal, bipolar, amacrine, or retinal ganglion cells. It is just the rods and cones. And so that is where our vision is sharpest. So that is where we have that best vision. Right, but again, this is a very, very small area, but this is dead center. So this is why you don't always have the most sharp vision if you're looking to the sides, but if you're looking straight ahead, and are able to focus that light on the fovea, then you're going to have much, much sharper vision. So here's that graphic. Again, we have covered the eye in lab, so a lot of these should be very familiar. But here's that fovea, so dead center directly behind the pupil. The optic disc and optic nerve are off center, as you can see here. And this is because of how the eyes are oriented and attached to the sockets. And then we have all of these different layers of that retina back here. So light comes in, and we've got that optic nerve layer of the retina from these ganglion cells. We have the amacrine cells scattered through here, bipolar cells right here, kind of in that center layer, horizontal cells deeper down, and then our rods and cones even further down from that. Right, and again, rods are sensing light, cones are sensing color. All right, so the rods and cones, this is where some issues can happen, especially with cones. So there are some genetic mutations that can cause issues with the function of the cones and lead to differences in color perception. So everybody's color perception is a little bit different to begin with, but then certain mutations can cause actual color blindness, so the inability to see certain colors. Red-green color blindness is the most common. This gene is linked to the X chromosome, and that is why people who have an X and a Y chromosome are more likely to have color blindness, is because they only need that one mutated copy of the gene on the one X chromosome that they receive in order to have that color blindness, whereas somebody who has two X chromosomes has to have that mutation on both of the X chromosomes that they have in order to be colorblind, because it is a recessive trait, so you have to have both of those copies of that gene in order to have that trait. If they've only got one copy and they've got the two X chromosomes, then the dominant one, the one that is not colorblind, is going to take over, and you will not notice that color blindness. There's also varying levels of color blindness, so there's kind of a step above that is color deficiency, where somebody might be able to distinguish between red and blue and, maybe like purple or something, so they can see those main colors, but shades of those colors can be really, really difficult for them. So one example, I actually had a student several years ago in my microbiology class, and one of the things that we do in microbiology is we artificially dye bacterial cells, and depending on the cell wall structure of those bacteria, some of them will stain purple and some of them will stain pink with a special staining process. And so a color deficient student, they had a very hard time distinguishing purple from pink because those are in that same color group of kind of those reds, just varying shades of reds with some different colors mixed in, but they were able to tell that what I saw as purple cells, they were darker than the pink cells, which showed up as much, much lighter, but they just weren't able to see that one was purple and one was pink. Whereas other people who are fully colorblind, those two cells could look very, very similar depending on exactly how much their color vision is affected. So there are some issues that can pop up with cones. There is also blue-yellow color blindness. That's a totally different mutation on an entirely different gene and different chromosome, and it is exceedingly rare. Very, very small percentage of the population has blue-yellow color blindness. Red-green color blindness is relatively common. I have had several students over the years in various classes that are red-green colorblind. Here's that fovea, different look at that fovea. So it is a depression because, again, we are lacking all of these upper layers of cells. We just have the rods and cones. All right, so that is where our vision is sharpest. And that wraps up our vision section. We will talk about integration of all of these different somatic pathways in the next video.